Antibody. This is the immunoglobulin. 20% of the total plasma proteins with the humoral immunity is producing the antibody. These are the glycoprotein which belongs to the immunoglobulins superfamily. They constitute most of the gamma globulin of the blood proteins. Rodney Porter and the Gerald Edelman revealed the structure and got the Nobel Prize in 1972. Large Y or T-shaped protein produced by the plasma to neutralize the pathogen is the antibody. Antibody recognizes the unique antigen via the FABs, fragment of antigen binding variable region. Each tip of Y of an antibody contains a paratope, which is specific to the epitope of antigen. Antigen binding site. Here we said the variable region. Variable region on both heavy chain and the light chain. The constant region on both heavy chain and the light chain. Structure of the immunoglobulin. An antibody, which is the immunoglobulin, that is a glycoproteins composed of one or more units, each containing the 4 polypeptide chain, 2 identical heavy chain and 2 identical light chain. The amino terminal end of the polypeptide chain shows a considerable variation in the amino acid composition. Each L chain consists of one variable domain VL and one constant domain CL. The H chain consists of variable domain and the 3 constant domain CH1, CH2 and the CH3, which contains 440 to 550 amino acid. Each heavy chain has about twice the number of amino acid and the molecular weight is nearly 50,000 atom, as each light chain have nearly 25,000 molecular weight, resulting in the total immunoglobulin monomer molecule weight of approximately 150,000 daltons. The heavy and the light chains are held together by the combination of the non-covalent interaction and the covalent interchain disulfide bond, forming a bilaterally symmetric structure. The V region of the H and L chain comprises the antigen binding site of the immunoglobulin molecule. Each Ig monomer contains 2 antigen binding sites and it is said to be bivalent. The hinge region is the area of a H chain between the first and the second constant region domain and is held together by the disulfide bond. This flexible hinge found in the IgG, IgA and IgD but not IgM or IgE region allows the distance between the two antigen binding sites to the vary. Hypervariable region of the hotspot present in both the heavy and the light chain variable region. The light chain variable domain include the 3 hypervariable regions present in between 23 to 24, 50 to 56 and 89 to 97 amino acid meiosis. In heavy chain, 4 hotspots present in between 1 to 23, 35 to 49, 57 to 88 and 98 to 107 amino acid meiosis. These are brought together to form paratopes. Paratopes, they are very specific to epitopes because of this. For example, IgG produced in response to different antigens show the variation in these hotspots only. Hinge region, it is a extended peptide sequence between CH1 and CH2 domains of heavy chain, rich in the cysteine. This hinge region, they are very rich in the cysteine and the proline AA amino acid, which is extremely variable in the amino acid sequence. It provides flexibility to the antigen binding part. Because of the proline content, hinge is highly vulnerable to the lytic enzymes, example papain and pepsin. Cysteine helps in the disulfide bond with the opposite heavy chain. Hinge is absent in IgM and IgE immunoglobulin. Instead, they have an additional constant domain which acts like hinge. Disulfide bond up. This is the single covalent bond between the thiol group of cysteine residues, also known as the S bond or the disulfide bridge. They play an important role in the folding and the stability of some proteins secreted to the EC medium. The total number of the disulfide bond depends upon the number of domains in the polypeptide chains of the IgG molecule. Carbohydrate meiosis. Significant amount of the carbohydrate in the CH2 region. It is a oligosaccharide with a variable monosaccharide. This may include the mannose as in IgM or N-acetyl lactoamidase as in IgG. No glycosylation in FAB region. The function is autonome. May help in the passage through the biological membrane. Causes of the immunoglobulin. The 5 primary classes of the immunoglobulin are IgG, IgM, IgA, IgD and IgE. These are the distinguished by the type of heavy chain found in the molecule. IgG have gamma chain, IgM have mu chain, IgA have alpha chain, IgE have epsilon chain, and IgD have the delta chain. Differences in the heavy chain polypeptide allow this immunoglobulin to function in the different types of immune response under the particular stage of the immune response. The polypeptide protein sequence responsible for these differences are found primarily in the Fc fragment. While there are 5 different types of heavy chains, there are only 2 main types of light chain, which is kappa and lambda. Antibody classes differ in their valency as a result of different numbers of Y-like units, which is monomer, the joint form complete protein. For example, in humans, the functioning IgM antibodies have 5 Y-shaped units containing the total number of 10 light chains and 10 heavy chain and 10 antigen binding sites. IgA is a dimer with 4 antigen binding sites. IgG, IgD and IgE are the monomer with 2 antigen binding sites. IgG. This is a monomer. This is a predominant Ig class present in the human serum. They are produced as a part of the secondary immune response to an antigen. This class of immunoglobulin constitutes approximately 75% of the total serum Ig. IgG is the only class of immunoglobulin that can cross the placenta in the humans. It is largely responsible for the protection of the newborn during the first month of life. Because of its relative abundance and the excellent specificity toward the antigen, IgG is the principal antibody used in the immunological research and the clinical diagnostics. The role of IgG in the immune responses is a major immunoglobulin in the blood, lymph fluid, cerebrospinal fluid, and the peritoneal fluid. A key player in the humoral immune response. Serum IgG in healthy humans presents approximately 15% of the total blood protein, the Fc protein of the IgG, but not FAB2 or the Fab fragments can cross the placenta of a mother and enter the fetal circulation, providing the fetus with a postpartum protection. IgG molecules are able to react with the Fc gamma receptors that are present on the surface of the macrophages, neutrophils, and the natural killer cells. They can activate the complement system. The binding of the Fc portion of the IgG to the receptor present on the phagocyte is a critical step in the opsonation. The phagocytosis of the molecules coated with the IgG antibodies is a vital mechanism that cells use to cope with microorganisms. The IgG is produced in the delayed response to the infection and can be retained in the body for the long time. The longevity in the serum makes the IgG most useful for the passive immunization by the transfer of this antibody. Detection of the IgG usually indicates a prior infection or the vaccination. IgG subclasses. There are 4 IgG subclasses. The subclasses differ in the number of disulfide bonds and the length and the flexibility of the hinge region. Except for the variable region, all immunoglobulin with one class share about 90% of homology, but only 6% among the classes. Determination of IgG subclasses can be valuable tool in indicating the potential antibody deficiency. IgG class, the molecular weight is nearly 1,50,000. Gamma is nearly 53,000 will be present. Serum concentration is 10 to 16 mg. Percentage of the total immunoglobulin is 75%. Glycosylation weight is 3%. Distribution through the intra and the extravascular fluid. Function is secondary response. IgM. The properties of the IgM is they are pentamers, largest antibody, molecular weight is 9 lakhs. H chain type, which is mu 65,000. The serum concentration is 0.5 to 2 mg per ml. Glycosylation weight is 12%. The distribution is they are present inside intravascular fluid. The function is the primary response. They eliminate the pathogen in early humoral immunity before the sufficient IgG. IgA class, they are dimer, secretory antibody, which the molecular weight is nearly 3,20,000 and serum concentration is 1 to 4 mg per ml. The percent of the total immunoglobulin is nearly 15%. Glycosylation is 10%. They have the distribution in the intravascular and the secretions like saliva, tears, breast, milk, mucosal areas of gut, respiratory tract, and the urinogenital tract. Their function is to protect the mucous membrane and prevent the pathogen colonization. IgD class, they are monomer, molecular weight is nearly 1,85,000 dalton, the half-life is nearly 2 to 3 days. The glycosylation rate is nearly 13%. They have the distribution in the lymphocyte surface. Their function is to an antigen receptor on the B cell which are not exposed to the antigen. They activate the basophil and the mast cell to produce the antimicrobial factors. The IgE class, they discovered by the Kirika in 1966, a scarce isotope found in the only mammals. The molecular weight is nearly 1,90,000 daltons. The half-life is nearly 2 to 3 days. The serum concentration is nearly 10 to 400 nanogram per ml. The glycosylation weight is nearly 12%. Basophils are the mast cells in the saliva and the nasal secretion. Their function is to protect against the parasite and the allergies. Functions of the antibodies, they help in the neutralization, agglutination, precipitation, opsonation, complement activation, activation of the effector cells and the natural antibodies. Neutralization, the neutralizing antibodies block the part of the surface of bacterial cells or the virion to render its attack ineffective. Agglutination, glue together, that is the foreign cells into the clumps. They are joined together. The clumps are the attractive agents for the phagocytosis. Precipitation, glue together serum soluble antigen, the forcing them to precipitate out of the solution in clumps. The clumps are the attractive targets of the phagocytosis. Complement activation, fixation in which the antibodies latched to the foreign cells encourage the complement to attack it with membrane attack complex, which leads to the lysis of the foreign cell, encouragement of inflammation by chemotactically attracting inflammatory cells. Natural antibodies, humans and the higher primates, which contains the natural antibodies, they are defined as the antibodies produced without any previous infection, vaccination, other foreign exposure, or the passive immunization. They can activate the classical complement pathway leads to the lysis of enveloped virus particles prior to the adaptive immunity. Rejection of the xenotransplanted organ is the result of the natural antibody. Thank you.